what is the function of these genes. Yeah. So we do not. There's an absence of screen, and of course earlier approaches have identified few genes that are important for cilia development function, yet not fully. And of course we have a whole repertoire of genes that are not yet characterized. So the goal in ciliopathy research then would be to identify new genes that are important for cilia structure function. Not only identifying these genes, uncovering the cellular mechanism of how these genes work, and then more importantly, how do we translate the knowledge that we acquire from such a study into advancing our understanding of human ciliopathies. In order to do all these things, you require a powerful model system where you can test what you want to test, and then of course validate these findings in a ciliopathy context. To this end, we utilize the zebrafish model. Zebrafish is an extremely powerful model system because it has very conserved genes, developmental and signaling pathways with human beings. It has, of course, very powerful genetics that can be exploited. And most importantly, you can screen a lot of embryos. So when you're out embarking to identify a whole host of genes, you need a system where you can continuously screen a lot of phenotypes, and you should be able to knock down genes very, very quickly. And that can be carried out at least in the fish model system. And of course, the ciliary structures are conserved. That's the criteria for it, because the cilia are not conserved from the fish to humans. It doesn't make sense in using the fish. Thankfully, it is conserved. And that's why the British Heart Foundation came up with this cartoon, which is not a fish, it is hope itself. Right. So at this point, our lab made a very landmark discovery in 2008. So where Jane Yu from my lab, what she found out is that there is this molecule that's called FOXJ1, or the fourth head transcription factor J1. She found that was the master regulator of motile ciliogenesis. What this means is just having this molecule is sufficient to make a motile cilia. So if you just ectopically activate FOXJ in a cell, you start the cell which previously never made a motile cilia will now start making a motile cilia, and this cilia can be. And likewise, if you remove FOXJ, so FOXJ loss of function, you start seeing that the motile cilia are absent, as you would see in the brain ventricles here. And this is just to show you that these cells which initially could not make a motile cilia now have started making a motile cilia. So this was a very important finding, because till then, to even realize that genes were important for making the cilia was a big thing. And now you had a gene that would make exclusively motile cilia. So what did we do? Then when I joined the lab, I asked, okay, so now that you have this molecule that can override the fate and make motile cilia, so what is it doing in the cell that is conferring the ability to make a motile cilia? So you have FOXJ1. And there are some intermediates that we do not know, and the final outcome of it is that we start having motile cilia. So then we did a whole genome microarray approach to find what are the targets of FOXJ. So what is FOXJ regulating in order to make a motile cilia? Right. So we did the experiment with 6 biological replicates. So you had the wild type, and you had the overexpression of FOXJ ectopically, and then this is the microarray of it. And what do we find? So in this plot, what you see here is that, so these are all various probes for various genes, and anything that's on this side indicates that it is overexpressed. So clearly, if you would just the visual impact of it, you would know that FOXJ is an activator, because you see a lot of probes are upregulated when FOXJ is ectopically activated. And what we find is that there's about 666 genes which are upregulated when FOXJ is overexpressed. And for us, it was important that we identify genes which are conserved from the fish to the human. So we were not interested just in fish-specific genes, but rather genes which have conserved roles. And from this 666, we find about 636 genes that mutate to mammalian orthologs. So this is the set we are interested in, what we call the ciliome set. Right. And as expected, so the 600, these genes, our list is definitely enriched for genes for functions which is ciliary or flagella motility, the cilium morphogenesis, left-right asymmetry, spermatogenesis, all of which have to do with ciliary development. Right. Now that we have this list of about 636 genes, how do we go further trying to dissect and making sense from that list? So what we decided, okay, so if we have this huge list, so we should be able to take any random gene from our list and any random cohort from our list and ask how much of these genes actually are FOXJ targets. So we selected about 55 random genes and did an RT-PCR validation. And then from these validated set, we went ahead to do a functional analysis, or a second screen, loss of function screen. And then parallelly also check where do these targets actually localize in the cilia. Right. So we had 95% positive rate. What you're looking at is the qPCR results for the 55 genes that we selected, or 52 of them turned out to be positive. So which means if we took any random set of genes, 55 genes from our list, we had about 52 of them which turned out to be positive for FOXJ, and which indeed is a target of FOXJ. So from this 52, we decided to characterize another 50 more. So we carried out a loss of function screen. So this was in essence what we call the reverse genetics approach. So what we did is we designed morpholinos. So morpholinos are these chemicals that can be injected into the embryo that actually blocks gene translation. So you have morpholino that was injected into a one-cell stage embryo, and then we scored for what is the result of such a perturbation of these. So what we assay for is actually defects in otoliths. I'll come to all of these one by one. Curved body axis, hydrocephaly, or development of kidney cysts. This is at the gross phenotype level. And at the molecular level, I asked how many of these have left-right asymmetric defects and what is happening to the cilia indicated on campus. Right. So the first phenotype, like I said, is the otolith. So what is marked here is the ear of the zebrafish embryo, and in this you start seeing these two particles. So these particles initially start out with clusters of particles, which are eventually eaten by the motile cilia present in the ear. So you have motile cilia that lines all along the surface here, and these particles then coalesce to form two structures that are called the otoliths, and these are thought to give balance to the fish. So when there is a defective motility of these inner ear cilia, what happens is that you start seeing these clumps of particles, you start seeing multiple otoliths, or even in cases you start seeing a single otolith. So from our screen, we were able to identify about 25 morphins which show greater than 10% otolith defects. So currently we are also live imaging to characterize these cilia repeat frequencies in them. So out of the 50 genes that we chose to knock down, we had phenotypes for 30 of them, which makes it close to about 60% hit rate in the screen. Then the next phenotype is the curved body axis. So here you see the wild type and you see the straight tail of the zebrafish embryo, whereas in these morphins, you start seeing that they start curving up. So it's still not clear why do these morphins curve up, but then this is a classical feature of a ciliopathy in the fish. So in fish models of ciliopathy, you start seeing that they start developing curved body by about 24 hours, which goes on to become severe by 50 hours. And we had about 23 such morphins out of the 30, which actually showed strong curved tail, so varies all the way from 10% all the way to 100%. Right. Then, like I said, the other phenotype is hydrocephaly. So hydrocephaly happens when the spinal canal is not able to, the cilia in the spinal canal is not beaten properly, and due to which there's fluid accumulation in the brain. So you can see that the brain ventricle here is fluid filled. So this is quite a severe phenotype in the sense embryos that have hydrocephaly generally will not progress beyond 3 or 4 days, and they die out by then. So these are all characteristic ciliopathy features, and we have about 23 of them, which again show hydrocephaly depending on from varying percentages. Right. And the other last gross phenotype is the kidney cyst. And kidney cyst is a harder phenotype to catch, partly because the number of embryos that will actually progress till about day 6 is lesser, and we've been able to catch about 6 of them which have defective kidneys. And you should remember, in the wild type and in the control injected morphinos, there are rarely any kidney cysts, and this goes true for any of these. What you see here, the wild type and the control do not have any hydrocephaly, they do not have curved body, or they have the control does not have any otolith, but then the wild type has about 4% of otolith defects. So all these defects that you're seeing are over and above, much higher than what it is in the wild type and control situation. Right. And the last one is like I said, the left-right asymmetry defect. So all of us, when we look at someone, it looks very symmetric, the left and the right side, but internally there is an asymmetry. And this asymmetry is decided very early on in life, at the embryonic state. So here we use this marker that's called the lefty 2, and you see in the fish embryo, lefty 2 turns on only to the left side. Whereas if there is a defect in the left-right asymmetric decision, you start seeing it either shifts to the right side, or it is bilateral on both sides, or in worst cases, it is none. There is no lefty 2 at all. And you see both in the control and in the wild type, it is 100% of embryos are on the left side, whereas this starts decreasing. You see the white bar actually starts going down in some of them and becomes very severe depending on the genotype. And now coming to the molecular, this thing, the actual ciliary length. So what is highlighted here is the structure that's called the pronephric duct. It's the kidney equivalent in humans. And in the wild type, you see thick, dense mesh. So this is a marker that's called acetylated tubulin that stains for cilia. And whereas in these morphins, you actually start seeing there is a reduction in length and reduction in numbers. In some cases, they become so severe that there are hardly any cilia left. And the other thing that we did is to ask where do these targets localize. To do that, we cloned all of these genes downstream of GFP, transcribed it from an SP6 promoter, and injected that into embryo. So here you see the GFP, the acetylated tubulin, which marks the cilia, and the co-localization for both. So we have about 20% of these genes which actually localize to the motile cilia. And these targets, some of them also localize to the primary cilia as well as the basal body, which is the base of the motile cilia. And so that is as far as the whole cilium was concerned about with 50 genes. In addition, we started doing detailed studies for about 3 candidate genes. One of them is LRRC gene. So you see there's a curved body and then there's a very strong reduction in the cilia. This is the wild type of thick meshwork of cilia. And there's very short stubs of cilia. So we've done an E2 hybrid with them and pulled out candidates which are important for endocytosis. The other gene that we have, so these are stories that I will not touch today for the lack of time, but we have about 3 candidate genes that we've done E2 hybrids and we are characterizing the interactions for all of them. Right. To sum up, so our screen has been successful in identifying genes involved in motile cilia formation. Like I said, we've had a 60% hit rate, which is quite good for a screen. And we've started to characterize the function of some of these genes, which I didn't have the time today to tell you about. And of course, pursuit of candidate genes from the screen will help identify positive mutations for several uncharacterized ciliopathies. And in parallel, what we started out as an EM project with NUH, where now we are characterizing these ciliary defects at the electron microscopic level for patients who have ciliopathies. So this is my lab, and that's my supervisor, Sudipta, and these are people who are involved in helping me in the project. And I'd like to end with the NGS theme, life, opportunities, and research. To me, cilia is a complex web, and it is the basis of life itself, be it in whatever way. And without the cilia, the question of continuity of life does not arise. And of course, it provides so many opportunities because there are a vast set of genes, molecules yet unknown. And of course, research, you can pick whatever your favorite model is, and there you go. So, cilia encompasses life, opportunities, and research. Thank you.
We just open the floor for maybe 1 or 2 quick questions.
You do better for J1 is also important in the biogenesis of the primary cilia as compared to motile.
Correct. No, so primary cilia are unaffected when FOXJ is knocked down. Yeah.
I'm also sure, because some of the genes are involved with the
Correct.
Looking at the kidney phenotype, only 6 genes.
Correct.
Good. Any of these resemble polycystic
Yeah.
So this is one.
Correct.
But these things are also important for primary cilia.
Correct. Absolutely.
Yeah, so you have a mixture of
Right. Right. And actually, the field is still yet uncertain about the distinction between primary cilia and motile cilia, because the easiest distinction will be at the length of it, or the fact that they don't have a dining arm. The primary cilia doesn't have a dining arm which confers a motility. But that apart, there are shared receptors on both of them, and it seems that the motile cilia can in part do what the primary cilia is doing, or likewise the primary cilia in part can do what the motile cilia is doing. So there's a conservation of functions.
There is a division of function.
Right. Correct.
Primary cilia is for sensory. So there are distinctions.
But then
there's very clear morphological
right.
you can do. It's a 9 plus 2 and 9.
9, yeah.
Yes.
9 plus 0.
So I cannot control you saying that there are.
No, so this was the traditional view that 9 plus 0, 9 plus 2, and then okay, these are receptors that present on the primary cilia, these are receptors on the motile cilia. But then increasingly what we are seeing is that the motile cilia, which is thought exclusively as a bit like this thing, is to beat around people, is no longer so, because especially in the neuronal cilia, motile cilia also have G protein receptors on them, which is exclusively thought to be the primary cilia has. So we're moving away from the traditional thought that this is just a primary cilia which doesn't move, and this is a motile cilia which only moves and just beats around, but then that view is changing now.
open the floor to
Is there
you.
any sequence that the phosphate will select?
Right, so FOXJ binding domain, you mean?
Yeah.
Right, so there is a FOXJ binding domain, but then these genes that are picked up from the screen, half of them do not have a FOXJ binding domain.
So these are two characterizations.
Right, you could say so.
Alright, so thank you, Deepak. I'm sure your findings will help us better understand the human stereopathy disease.
And so now we transition from the stereogenesis pathways to understanding the dynamics of elastic solid-solid interactions. Let us now invite Abouzar Kaboudian on stage to tell us more. Abouzar, please.
Okay, hello and good morning, everybody who's still here in the morning. Okay. Thank you for all the previous presentations. Today I'm going to talk about closed-solid method algorithms for solid-solid interactions. I try to keep the jargon to the minimum so that nobody will try to shoot me here. Okay. I start with a short introduction of what I'm going to talk about, and then I will try to explain this method for one-dimensional case of waves through solid-solid interaction. And then I will show through example how when this method is extended to a two-dimensional case, it will behave. And then I will have a short conclusion, and I will talk about if there are any future works possible, and then I will answer any questions which may arise. Okay. So, dynamics of solid-solid interactions can be useful in various areas. For example, geophysical dynamics, where you can see different layers of sand are available. And if there is any wave traveling through these layers of sand, they will start interacting with each other. And you have to be able to take care of these interactions. Or when we are having some non-destructive testings in engineering. So you send some sound signal, acoustic signals to the solid, and then you read the feedback to understand whether there is any defects in the solid. So if there are many layers of solid, then you have to take care of it, be able to take care of that. Or in biomedical research, where you obviously have so many layers of solid, for example, muscles, and then you have some bones underneath you may have, and many more fields that solids interact with each other. So there are different approaches to tackle this problem. First approach is by doing some experimentation. Of course, you have to prepare some samples. These samples take time to be prepared, and then you have to set a probe to do the tests, and finally in the end, if the test is not okay, is not satisfactory for your design, you have to start all over again. And this process is both time consuming and also very expensive. And whatever results you have, you cannot just transfer it to any other geometry. The other approach is an analytical approach. You start with pen and paper, you start writing down the equations, and reach at a solution. But this is not possible for complex geometry and also complex boundary conditions. The third one is the numerical approach. You write a numerical code, you develop a method, and then you do a simulation. All you need is one computer or just a bunch of computers to solve your problem. It's very easy for both complex domains and simple domains that you may have. And also can be easily modified. If you are not satisfied with the results, you can change the geometry and just run the code in. And it's very inexpensive. But the downside is that it can suffer from numerical errors, meaning that the results may not be very reliable. So there should be other methods to verify that these results that you are obtaining is actually correct. So we compare it with analytical results or some experimental data. So what we use here is the numerical simulation methods. So when you start doing numerical simulations, you have to take some steps. First, you have a domain. Here you have a simple rectangular domain. And then you want to start doing some simulations. The first step is to discretize time and space, because you cannot find the answer for all of the areas of this domain. You take some sample points. It's just like putting probes on each of these points, and then get the results for each and every one of these points. And then also for the time, you have to discretize the time. For example, for every 1 second, I find the answer. When I have discretized the time and space, I start solving the problem. I make a computational cell, meaning that I use, for this example, these 4 nodes or these 5 nodes to find the answer, this middle node. And I do the same thing for all these nodes at each time step, so I can find the answer. And once I have the answer, I have to post-process it, I have to show it something. So in the middle, there has been several methods, several algorithms developed for single solids. Throughout the whole years, for 50 years, people have been developing methods for single solids. The problem is that then you have 2 solids, and the boundary of these 2 solids, the interface passes through this computational domain. You cannot simply assume that you can use the previous computational cell, because the material properties on the both sides of the domain are different. Moreover, there may be some slip at the interface, so the computational domain may not remain the same after some time. So you have to be able to take care of this problem in an efficient way and also in an accurate way. So what I'm trying to do is to take care of this interaction with minimal change to my original code, which is for 1 single solid. So I start with a 1D simulation, because for 1D, first of all, analytical solutions can be easily obtained. And also, the numerical solutions are easier to formulate. And also, the numerical schemes obtained for 1D case can be extended to multidimensional cases. And also under certain simplifying assumptions, I can change my 2-dimensional cases, some of my 2-dimensional cases to 1D problem. The outline of the problem is like this. This is a 1D domain. Just imagine that I have 3 nodes here and 3 nodes on the right-hand side of the interface. 1 solid on the left-hand side, and the material properties of this are different. What I do is that I assume that there are some nodes, ghost nodes on the right-hand side, which together with the nodes on the left-hand side just make 1 medium. So my 1 single medium code can solve for all of these nodes. These ghost nodes do not physically exist on my problem. I define them to be there just to fit the numerical code so that I can easily use the numerical code. Depending on how I define these ghost nodes, different methods of ghost solid methods can be obtained. For example, the simplest one, the original ghost solid method, is that I take the material properties of this point and copy it to these ghost nodes, meaning that I, for example, for the density or the speed of sound in this material, I just copy to the ghost nodes. And for other properties like velocity or the stress or pressure, what you may call it in 1-dimensional case, I just copy from the real nodes that are there. And then I solve for medium 1. I do the same thing for medium 2. Instead, I have some ghost nodes on the left-hand side. So I solve with a single medium solver, just 2 times for each time step. 1 for medium 1, 1 for medium 2. I don't change the code at all. I just define these ghost nodes. The modified version is a little bit different. The material properties I copy to these ghost nodes, but instead of taking the velocity of these points, velocity and pressure at these points, and copying to these ghost nodes, what I do, I copy from the interface. So whatever values of velocity and pressure I have from the interface, I copy to these ones. I do the same thing for the left ghost nodes. When I perform some numerical experiments, the results are like this. The interface was at the middle, at 5, and you can see that these green dots are the original one that I simply copied from the real nodes to the ghost nodes. You can see, and the blue line, which is hardly visible here, is the exact results which are obtained through analytical solution, is this line. Okay. These are all scattered all over the place. These are numerical oscillations, meaning that around this area, my answer is not correctly the original one. But just by changing it from copying the interface to this ghost nodes, I get all the results following the exact solution. And you can see for the velocity profile, the same problem exists with the original method, but with the modified one, I am easily following the velocity profile. If I start doing the 2D modeling, the results will be different. Why 2D? Because 1D has very limited practical application, almost no practical application. And developing 2D models can show how I can extend to 3D, multidimensional cases. So I start with a very simple problem. There is a left solid here, there is a right solid here, and there is an interface in between. Once I do the simulation, I see that the original ghost solid method gives the oscillations here, but the modified version removes all these non-physical oscillations. Also, for the stress on the other side, you can see the steel, these oscillations are present. If I plot the results in the mid-plane for better visualization, the original ghost solid method leaves these very disturbing oscillations, and the modified ghost solid method gives a very nice profile of the method. So the conclusion is that the original ghost solid method is very simple. You just copy from the real nodes to the solid nodes. You don't need to solve any problem for the interface to find out what's the interfacial values. And it can be easily extended to multidimensional cases. But the problem is that it's very much problem related. For this critical case, it was shown that it can blow up and the results are totally not reliable. And it can lead to non-physical oscillations. While the other version, the modified version of this ghost solid method, is much less problem related. It eliminates these oscillations which arise due to this one, and can be successfully applied to elastic solid-solid interaction problems. However, you need to find that interfacial value to copy to the ghost node, and it can be much more complicated when you move to a 2D problem, because you have to find these interfacial values accurately. And how do you copy from these interfacial values to the 2D problem is a little bit complicated. For future works, you can do some error estimations of these GSM-based algorithms. We can develop GSM-based algorithms for other frameworks, and we can also develop a systematic method to properly define the material properties over the ghost nodes. Right now, we are just simply copying from the left-hand side to the right-hand side the material properties like density or the speed of sound. We can modify this one to avoid any possible oscillations that may arise. And we can apply these GSM-based algorithms to real-life problems. And we can also develop GSMs, meaning ghost solid methods and ghost fluid methods for viscous fluid and solid contraction. In the end, I wanted to thank my supervisor for his guidance throughout this research, and also my co-supervisor, Dr. Jong Hu, and all the participants. Thank you.
Is there any questions? We have time for one or two.
Actually, I was going to ask about it, but you kind of showed it in your last slide. I was going to ask if this method should be extended to solid liquid interfaces, because I was thinking back to what Jeffrey presented a bit earlier on, the liquid interface project, and I'm wondering whether such algorithms could be worked.
These algorithms have been already used for fluid-solid interactions, but for inviscid fluid, meaning the fluid is considered to be non-viscous. So the possible future work is that to take care of the viscosity, because normally nowadays people, when they are talking about fluid-solid interaction, they consider the fluid to be inviscid because it's mostly aerodynamics, and they can easily ignore the viscosity of the fluid. But there are cases that you cannot easily ignore it, and you can. Any other questions? Yes?
So, do you have like an intuitive explanation for why it makes such a big difference whether you copy properties of the liquid instead of the
It's because of the governing equation itself, and also the fact that once we are calculating these numerical differentiations, when you are just simply taking the values at these points, you may have a big jump at the interface. And this big jump means that once you are calculating, for example, the value of some node and minusing the value of another node from the other side of the interface, and dividing it by the cell size, the computational cell size, this value can be very large if there is a jump. And once you make this grid smaller and smaller, this delta x in the denominator will be very small while the value above is still large. And it leads to very severe oscillation. But when you are copying the exact value of the interfacial values, you introduce a little numerical viscosity. And also you copy the values which are more close to the values on each side.
I didn't really quite understand. You showed these oscillations, but
Yes.
if you have a wave interacting with a surface, you'd expect to get reflections and then the same wave setup. So there will be oscillations.
No, there is physical oscillation and there is non-physical oscillations just due to the numerical code. So these oscillations that I showed are just numerical oscillations which do not exist in reality. So what you are going to do is that you want to have a code that can take care of the physical oscillation but is free from numerical oscillations. So what you are reading or predicting is actually correct. What I was showing that this method was suffering, which was the extension of a previous work, the original post-solid method, suffers from numerical oscillations, which are actually wrong answers.
So can you deal with evanescent waves?
Yes.
And your model doesn't take into account shear waves.
It does take care into account shear waves for the 2D case. There are different boundary conditions that I didn't show here in this presentation. There are two cases. Once you have, you allow sleep at the interface, or you don't allow sleep. So there are two different cases, and there is one in between that there is sleep, and also there is shear. So each of these boundary conditions, the original work cannot take care of these ones, but the modified one, once you take care of the interfacial values, you actually solve these ones, and then you copy into the other one. So you take care of the shear waves as well.
All right, thank you, Abouzar, for your interesting talk. I'm sure
Thank
your
you.
algorithms will have potential impact in real-life application, especially in three-dimensional space.
Thank you very much.
So we have heard how hair-like organelles are formed and how solid interacts with solid. Now let us find more about the molecular mechanisms of our brain and how it translates to an outward expression of behaviors. Let us now invite Shawn Tan to give us his talk before our lunch break. Shawn Tan.
Sorry, can you hear me? So, I find it interesting that Prof. Dave Derrick talked this morning about what real interdisciplinary research should be, and I'm not sure if I'll be among one of the charlatans doing bad science. So, I'm actually a first year NGS student. I haven't really embarked on my research yet, so I'll leave you all to decide whether in the future I'm doing good work or not. So, I come from psychology. It's a behavioral science, and I work a lot with people. And as a neuroscientist, I'm interested primarily in this question: how does the brain work? Now, the people who have tried to answer this question have been divided into two camps. The molecular or cellular neuroscientists believe that the brain could be understood as a complex mix of chemicals, and that if one knows what certain proteins do or what certain neurotransmitters do, one can understand behavior. The systems neuroscientists, which I come from, believes that you don't need to go down to that level, and that one can make certain generalizations or predictions about how different areas of the brain contribute to complex behavior. So, in order to understand the mind control techniques that I'll be presenting, and why exactly I think that they are so exciting for neuroscience, we first need to learn a bit about the history and the limitations of the various techniques that we use. So, most of the systems neuroscientists, the gold standard they use is this method called fMRI. Many of you have no doubt heard about this technique. And basically what it allows one to do is to image the neural activity in a normal individual while he is doing some normal behavioral tasks. This method has really revolutionized the field because it's non-invasive and in fact it's the only method so far which can be done looking at neural circuits in humans. However, if you look at the fMRI, there are several problems, and these are problems that people don't really like to talk about. So imagine that you have here this high definition image, and it's an actual snapshot of all the activity in the neurons in the brain at a single moment. This is the image that you see under fMRI scan, and you notice that immediately the image is quite blurred, and that is because even though the number of voxels in a typical fMRI is about 150,000, there are actually about 100 billion neurons in the brain. So each dot in that picture is actually trying to represent the activity from over 600,000 neurons. And, sorry. Okay. And another thing is that neurons actually fire many times a second, and this information is actually valuable, but the fMRI can only average activity that happens over a short period of time. The impact of this loss of information becomes clear when one looks at what neuroimaging studies can tell us. So you have here an area of the brain called the orbital frontal cortex. It lies just behind the eyes, and it has been shown to be implicated in many things such as the integration of different things like vision and audition, or regulating emotion, understanding social cues, computing the value of rewards, and complex decision making. So, why one area can serve so many tasks is because there are actually many diverse neurons in the brain, and this is just a representation. It shows 21 different classes of a type of neuron called the interneuron, 3 different pyramidal cells, and this is just coming from one area of the hippocampus. So, okay, let me just go back. So, the the people who are studying brain areas have faced a roadblock in terms of the amount of information that they can get just by looking at a very broad level. So if you switch over to the molecular side, the molecular people are actually somewhat better because they have quite a few tools at their disposal. And I'll just go through 2 of them. So I think the biggest thing for the molecular people is the gene revolution, which has enabled the molecular neuroscientists to knock out specific proteins from an animal and to compare it to a non-knockout animal. And this shows that these mice, when a gene is knocked out, it becomes obese. One can also do pharmacological interventions. So instead of knocking out a single protein, one can actually use drugs to try and target and block the action of certain of these proteins. And one can actually even do a bit of a targeting. But although the gene knockout and the pharmacological approaches have enabled us to understand and elucidate some of the mechanisms behind different genes, what one realizes is that actually many cell functions are mediated by the same targets. So this is one example why whenever you take a drug, you have a multitude of side effects. And this is just an example of in the treatment of depression, most of the drugs try to elevate the levels of this neurotransmitter called serotonin. The thing is that serotonin receptors are found throughout the brain. So when you try and use these drugs, you have things like sexual dysfunction, suicidal behavior, fatigue, weight loss, and the list goes on. So the molecular methods can tell us certain things, but it cannot really dissociate really complex behaviors. So at the end of the day, in order to understand both camps have now realized that you need a certain level of integration and information for both levels. In order to understand neural circuits, you need some cell-specific or molecular information. And in order to understand molecular and biochemical imbalances, you need to understand them in terms of where they are, where they come into play, and how they affect the signaling of neural circuits. So the question is, what tool can offer this kind of information at different levels? So the tools that we're talking about are actually one subset. There are actually many tools coming into the field right now that can allow one to get information at these different levels. But I think these mind control tools are better in a sense because they allow one to experimentally manipulate certain conditions, and they work on several similar principles. So most of them can differentiate between different sets of neurons in the same area of the brain. They can manipulate them while keeping the rest of the brain intact. And you can also switch them off after. I think this is very important when one tries to understand how different neural circuit signaling leads to complex behaviors. So this is a tool that has garnered a lot of attention, and deservedly so. And I think the principle is basically quite easy to understand. So I'm going to try and bring a bit of how it works inside. So you have this gene for a light-sensitive protein that's coupled to a cell-specific promoter. When this is packaged into a virus, which delivers this protein to the neuron that you want, a protein is produced that responds specifically to light. And when you shine light on this protein, it will actually open up and it will depolarize the membrane or hyperpolarize the membrane, thus leading to neuronal activation or deactivation. Okay, and here's an empirical demonstration of this. This is sound of this. So basically, I'll just explain. So the mice has this fiber optic cable that's transmitting blue light to this to a subpopulation of neurons, which controls the movement. And you can see at first, when the blue light is not turned on, it's acting normally. But you can see there's a blue light, that's being turned on now, and it starts to move around in a circle. So this technique, as you can see, is very time sensitive and it's very sensitive in the spatial dimension as well. But it can be a bit difficult to get light to certain areas. Basically, you have to thread a fiber optic through, and that can damage the areas which you do not intend to manipulate. The second set of tools which I'm currently working on, and I think have been neglected somewhat, are the chemical genetic tools. And the principle is basically the same, but instead of being activated by light, these proteins are activated by a drug that has no effect on in the body, has no natural effect in the body. So when you activate this protein, it does basically the same thing. So this is slightly better in that you can penetrate deep brain areas, and it's also more convenient in that in order to control the animals, you don't need to use any complicated fiber optic signaling, and all you need to do is to just give the animal a drug. And what is not shown here actually is that this method uses natural signaling pathways that are already present. So one could do various biochemical manipulations, and this will be very useful for the molecular people. Oh. The last technique and it's the most is the one that is my favorite because of how audacious it is, and just the mad science that goes behind it is the magnetic nanogenetic method. And basically what you have, the same principle, is a protein that's activated by heat. But the question being, how can you deliver heat to neurons without frying the rest of the brain? And so you have an anchor protein onto which nanoparticle is attached, and when you deliver magnetic radiation from outside, these channels will open and the same thing happens. So this combines the advantages of both methods in that it can penetrate deep into the brain and is time sensitive, but as to how applicable it is and how easy it will be to implement is something that needs to be worked out. And this is a new method, it's just like 2010. And so just to give you a real life example about how this method can help. So people who are investigating the neural basis of fear and anxiety have identified this area of the brain called the amygdala that is responsive when one is in a fearful situation or when one feels fear. However, it's quite dissociate between what different areas of the amygdala are doing in different fear processes. On the other hand, you can see that there are various drugs that can reduce anxiety behaviors, but they also affect other parts of the brain. And there is one protein knockout mice that has less anxiety, but this protein is actually a microtubule protein, and is actually highly expressed in many different areas. So these mice also have significant behavioral deficits. And with some of these optogenetic tools, you could target different areas of the amygdala separately, or you could target a specific subset of neurons within the amygdala. And these people who have used this method have found that a subset of cells actually act to block the expression of fear. And so I believe that these techniques hold great promise, but they're not the only methods that are available. And I think what's happening in the field of neuroscience is that it's actually moving towards a convergence, a point where you need level information at the cellular level in order to understand molecular and systems level problems. So for those of you all who are not neuroscientists, you're here thinking, okay, so this is great for the brain people, but what does it do for me? And actually, the thing is that these tools are constantly being refined to control different cellular events. And so in this case, you can see that they are using a laser to control the movement of this skin cell. And one can think of things like paralysis, where basically the spinal cord nerve connections are severed, and people have been trying to use stem cells to grow these connections back, but they haven't been able to get the nerves to synapse properly and to form connections, and they haven't been able to get them to migrate specifically. So this can help in nerve regeneration. And so I leave you with this quote from Caldas Ruff, who is one of the pioneers of the field. And one can certainly think about the various applications for different things beyond neuroscience. Okay, thank you, and I hope to have something more concrete for your next year. Okay.
We're open the floor for 1 or 2 questions. Shawn?
I have a question.
Yeah.
I'm just curious. I've explored certain aspects of this research as well, and I'm more curious with the magnetic nano stabilization. So in our laboratory, we are currently trying to conceptualize a way that we can transmit over the network the sense of smell. So currently what we're doing is that we're looking at using magnetic fields for concentrated magnetic fields for deep brain stimulation. But the problem that we have is that the area of the brain that smells is actually quite deep, and in order to use a concentrated magnet, we would probably damage a lot of the brain going down to that. So I'm just curious if you could educate me, being a designer, and tell me, would it be possible to use the nanoparticle technique to simulate smell? And would we be able to use that as, say, a communication medium in the future?
So I need to understand several things. So the magnetic stimulation that you're doing right now, is there any way to focus or target?
It's all theoretical right now, so we haven't actually built any devices to do that, because first of all, IRB probably wouldn't let us do that.
Okay.
But, so we're more or less trying to teach ourselves the fundamentals of neuroscience at this point.
Okay. Well, there are actually, so you've heard of transcranial magnetic stimulation?
Yeah.
I think the hardest problem is trying to get information out of deep brain areas. And while these techniques can allow you to manipulate, they're not so good at getting the information out, right? And that's the...
Well, see, when you say information, I'm not necessarily saying like I taste a red wine in Paris. I say, wow, this is great, I want you to taste that. So I send you a smell or a flavor SMS to your brain. I'm not saying quite so literal. I'm just thinking, is it possible in the first place? So the message does not need to be a literal message. It can be maybe a poetic message where your interpretation of it is completely different. So when I talk about when you say information, I mean actually any information.
Yeah. So I think you could use basically the olfactory senses. I think there are certain cell-specific promoters that you can use to identify the neurons in the brain that are responsible in some aspect of smell, and you could certainly manipulate them or try and stimulate them to give rise to the perception of the things that you're talking about. Yeah.
You've got to start trying with it. It's a really interesting question, but given that work that suggests that if you put a series of electrodes on a blind person's tongue, which then connected to a camera, you can get them to see. Yeah. Not the way we see, but they can actually see something. And that suggests that these fundamental senses are clearly connected. So why not transmit something visual and then actually have from your iPhone tongue adapter?
Yeah, so there's a whole, that's interesting because in HCI, human interaction, there's a whole emerging field called, I can't remember the term when you see something but you taste. So there's a mixing of the modalities of your senses. Synesthesia.
Yeah.
So there's a whole research field emerging research field called synesthesic media. So how can we make media that translates one sense into another? And so that's why, I know nothing about the brain that you're describing to on your level, but I think you could really come into our lab and teach us a lot. So
I'm certainly looking, so I come from psychology, right? And the reason why I put this image up is that ultimately, I'm not so much interested in the molecular stuff so much as to how they give rise and how they can control behavioral and cognitive phenomena. So certainly if, the thing with these techniques is that I've neglected to mention, perhaps somewhat purposefully, is that you can't really use them on humans yet. So in a sense they are quite limited, but technically they are reversible and they are safe. It's just that even I wouldn't want to put myself under it, so how can I ask someone to... so if you're thinking of trying to send or trying to use these techniques to cross vision or sensory or auditory modalities, the question is, what can you do it in?
So that's a great question because yes, we think as human beings that's something that we would want to replicate possibly is transmitting a smell or a taste. What if we could simulate that in a computer simulation and put it into a robot? So another project in our lab is that we've essentially computer modeled with using AI techniques the human endocrine system. So I'm just thinking if it's possible to kind of create like a cybernetic smelling system using some of these technologies, like concepts from these technologies.
I could imagine theoretically that you have this field of human neurons that have been harvested from a certain perhaps dead person or perhaps grown in the lab, and one could use these techniques to using light to control these circuits. And theoretically one could build a cybernetic robot with human neurons and with these things to control various aspects of it.
That's awesome.
That's awesome, right?
Yeah,
it sounds really great, but moral and ethical issues. Yeah.
Thank you.
Okay.
Thank you, Shawn, for the interesting talk. I'm sure you don't want to be like the dog, Especially don't show this slide for your oral exams.
One more thing is to always use your personal protective equipment. Okay, thank you.
So ladies and gentlemen, while our minds have been well fed with information, our stomachs need some attention too. A nice lunch spread has been prepared for you outside, and we will reconvene here at 1:30. So